So continuing on with the muscle section that we started on Tuesday, some of this will be a review from Tuesday. So starting off with looking at the contractile proteins that on the very, very smallest level are what actually produce the muscle contractions. So there's two types of contractile proteins that we see. We have thick and thin filaments. Does anyone remember what they're called? Awesome. Actin and myosin. Okay, very good. Actin is the thin filament. Myosin is the thick filament. Actin and myosin are arranged within a functional unit called, does anyone remember that one? Sarcomere. Nice. So actin and myosin are arranged in a functional unit called a sarcomere. We're going to look much more closely at the sarcomere today and closer at actin and myosin. So actin is the thin filament. All we really said about it on Tuesday is that it's the thin filament. But if we take a much closer look, we see here's a strand of actin right here. It's made up of filamentous actin, or F-actin. If we were to break down the pieces of the actin, we would see that we've got a few different things in here. So first off, we've got these green balls. This is the G-actin. The G-actin here. On the G-actin is where we find that binding site that myosin likes to attach to. So recap from Tuesday, myosin has that head that likes to grab onto actin, but it can only grab on to a special binding site. So looking all the way zoomed in at actin, we see that that binding site specifically is located on the G-actin, on the little green balls. What else do we have on actin? Well, we've got tropomyosin, that's this kind of purple colored rope here, if you will. And we've got troponin. We talked about those two on Tuesday also. The binding site on actin, where myosin likes to attach to, that binding site is covered when we are not actively using our muscles. It is covered by tropomyosin. Tropomyosin is held in place by troponin. Troponin is the little yellow balls. So tropomyosin covers that binding site when we are not using those particular muscles. And troponin kind of locks tropomyosin into place. Now, if we look at troponin, troponin has three subunits. So this is the troponin complex here. But we can see there's three little balls. So there's three pieces to it. Each piece has its own special purpose, and this makes sense when we think about how actin works. So the three subunits, one subunit binds to the tropomyosin. This makes sense because troponin is what holds tropomyosin in place. So it would make sense that one of the pieces binds to tropomyosin. The other one binds to G-actin, the green balls. This also makes sense because troponin binds tropomyosin in place. Well, it has to attach to something on the other side, so it attaches to the G-actin. That's how it keeps it in place. The third subunit binds to calcium. This is something we haven't talked about yet, and we'll talk about a lot today. calcium plays a very important role in muscle contraction. We need calcium to come in, bind to troponin. When calcium binds to troponin, it changes the shape. As the shape changes, it pulls tropomyosin off of that binding site and leaves it exposed, allowing myosin to come in and attach. So I'll say that again. We need calcium for muscle contractions because calcium comes in and binds to troponin. It has a special spot on troponin. When calcium binds to troponin, it changes the shape. When the shape of troponin changes, because troponin is bound to tropomyosin, it pulls tropomyosin out of place and exposes that binding site on actin. One of the video clips I'm going to show you will illustrate that. Okay, good so far? Okay, good. Now let's take a look at myosin closer. So myosin, this is our thick filament. It has about 300 molecules of myosin. And a molecule of myosin includes heads and the tail. The heads are the portions that attach to that binding site on actin. Once they are attached, that forms that cross bridge. We'll go over that again today. And then pulls actin towards the center of the sarcomere, which is the actual contraction part. So the head attaches to the active site, that binding site on actin. Okay? Now we're going to take a closer look at the sarcomere. Essentially, we're just going to be naming the different zones and sections of the sarcomere. Now again, the sarcomere is the functional unit, the smallest functional unit. And it's the functional unit because actin and myosin just kind of randomly by themselves don't do anything. They need to be able to interact. And the way they're overlapped is important because it maximizes how much interaction occurred between the actin and the myosin. So here is one way of depicting the sarcomere. The thin lines here are going to be actin. The thicker lines here are going to be myosin. And we can name the various zones. So we have A bands. The A band is in the center. The A band includes everything here. So the A band has actin and myosin in it. The very center of the A band is called the H zone. Do I have that on this slide? Yes, the H zone. What protein filament is found in the H zone? Yeah? Yeah, good, nice. Does everyone see that? The H zone is the very middle. So if we're looking at only this section here, the stuff in between my fingers is just the myosin. Yes?
I'm sorry, is there a reason to split H zone for all these letters actually?
Say that again?
Is there a reason that these particular letters are used?
Yeah, because we have to name absolutely everything. Yeah.
Well, it's just something for logic, something for conversions.
Oh, like why is it called the H zone?
Yes.
Ooh. I don't know actually. I can look it up and see if there's like an interesting reason, but off the top of my head, I don't know. So H zone, very center, includes just myosin. And then on the sides, we have the I band. Sorry, I can't remember what I have on which slide. On the sides, we have the I band. Which protein filament is found in the I band? Actin. Okay, so everyone sees that. Okay. On the exam, I will have questions specifically about the sarcomere and about what you can see and what resides in each section. Okay, the very center of the H zone is bisected by an M line, which you can see in this picture here. This is essentially where all the myosin is tied together in the very center. And the H zone is, the A band is the zone of overlap. The zone of overlap is very important in the sarcomere because this is the area where the actin and the myosin are stacked on top of each other, and this is where the interaction is possible. So if we were to take a cross section, so like cut it this way and then turn it and look at it from straight on, this is what the zone of overlap would look like. So the thicker red dots are showing you myosin, and the smaller green dots are showing you actin. So each myosin is surrounded by 6 actin, and each actin is surrounded by 3 myosin. This is important because the more chances actin and myosin have to grab onto each other, the more cross bridges we can make, and the stronger the muscle contraction we can produce. So that occurs in that zone of overlap in the A band. And then the I bands on the outsides here are bisected by the Z lines. And the Z lines are another anchor point where essentially the actin filaments are tied together. Then we have this elastic filament called titin that essentially anchors the myosin molecule to the Z line. So we don't just have actin and myosin kind of floating around there. They are anchored in place. They are attached in the middle. They're attached on the ends, and all the pieces are situated in their perfect spot. This allows for the sarcomere to shorten during contraction and then return to its resting state without getting out of order or being arranged poorly.
I think I found why the different letters are used.
Oh, okay, yeah, what is it?
It's German. The Z line is zwischen, meaning between. A band is for, I band is for isotropic, A band is for anisotropic, H zone for heller, or brighter.
Oh, brighter, that does make sense, actually.
M line is just, it's the middle.
Middle, yeah, that's good. Oh, thanks. Okay, this starts the complicated steps of what is exactly involved in muscle contraction. So what I'm going to do is give you like a very basic summary of what we're going to talk about on this slide. Then I'm going to go through each point step by step. We'll read it, and then I'll translate it into something that makes sense to you. So what we're looking at now in the next several slides is what exactly precisely happens when we activate and use our skeletal muscle. So in the big picture, what's happening is, and you've probably heard me say this a few times already, when we're going to use our skeletal muscle, because it is voluntary, this begins with a thought in our brain. That thought is actually an electrical impulse. We can also call that an action potential. That's what you're going to see on these slides, is action potential. That action potential is sent from the brain down the spinal cord, from the spinal cord to whichever correct nerve is going to talk to whichever muscle we want to innervate. Then that action potential physically travels into the muscle and allows the muscle to contract. I'm kind of glossing over some detail there. This first slide here, all we're looking at in this first slide is that electrical signal, that action potential, traveling down and arriving at the muscle. That's what this whole slide is. So what we're going to look at are the steps of the action potential traveling down the nerve and arriving at the muscle. So this is the signal that tells the muscle to work. Good so far? Yes?
Is the ACH, is that actually recorded?
Yes. All right. So step one, action potential travels down an axon of a motor neuron to the synaptic terminal. Okay. We haven't looked at the nervous system yet. A motor neuron is just the name of a nerve cell that talks to muscles. Okay? The axon is just the name of the physical pathway that it takes, like the little highway. So the action potential, that signal, travels down the length of the nerve and arrives at the synaptic terminal, is just the end of it. Like when you get to your terminal at the airport. From that terminal at the airport, you're going to go somewhere else. So when that action potential reaches the synaptic terminal, it's reached the end of that motor neuron. The next thing it has to do is reach the muscle. In this picture here, this whole thing is our motor neuron. It's the nerve cell. The little lightning bolt is showing you the action potential. So that action potential travels down the motor neuron and arrives at the synaptic terminal. The synapse is just the name for the space between the neuron and whatever comes next. The synapse is this space right here. The synaptic terminal is just the end of the motor neuron. So when that action potential travels down the motor neuron and reaches the synaptic terminal, it is going to change the permeability of the membrane. So the membranes, they're wrapping around the cell. Changing the permeability of the membrane means that we are now going to let some more things in and out of the cell. Okay, now we'll look at step 2. ACH is released. ACH stands for acetylcholine. Acetylcholine is a neurotransmitter. We use neurotransmitters for our nerve cells to communicate. Whenever we want to send a message, we use a neurotransmitter. So ACH is released into the synaptic cleft, it's just the open space here, at the neuromuscular junction. Neuromuscular junction is just a fancy way of saying neuro for neuron, muscular for muscle. Neuromuscular junction is where a neuron meets a muscle. That's all that is. So far, yeah.
So what did you say?
Acetylcholine. The video clip I'm going to show you has captions on the bottom, and it, I think, says acetylcholine on it, because I don't actually think I have it written. Okay, so far what we have is the action potential has traveled down to the end of the motor neuron. It's reached the neuromuscular junction. The permeability of the membrane has changed. So we're seeing, this is acetylcholine here, we're seeing it leave the motor neuron and it's crossing the synapse. So next step, ACH, acetylcholine, binds to receptors at the motor end plate of the muscle fiber. The motor end plate of the muscle fiber is just the name of the spot that has the receptors. So we have the neurotransmitter acetylcholine has left the motor neuron, it's traveled across the synapse, and it's going to fit into the receptors on the muscle fiber. So at this point, the muscle fiber has now received a signal. This is the communication. Okay, so the motor end plate is a highly folded section of the sarcolemma where we have the receptors. Once that acetylcholine reaches its spot in the receptor, it starts being broken down. It's broken down by an enzyme called acetylcholinesterase. I know, who names these things? Anytime you see ACE, ASE at the end of a word, that's a clue that you're looking at an enzyme. So then the word does make sense because acetylcholinesterase breaks down acetylcholine.
What's the ester? Is that ester or ester?
Acetylcholinesterase?
Yeah, it just doesn't E there.
There isn't
Well,
either.
anyway, the ester means that it breaks down something.
It could. You're quite the linguist. I'd have to look that one up too. So acetylcholinesterase is the enzyme that starts breaking down acetylcholine. Okay. So all we have up until this point, the action potential has been sent down the motor neuron. The neurotransmitter acetylcholine has crossed the synapse, has fit into its receptors at the motor end plate on the muscle fiber. The enzyme starts breaking it down. Now, as the result of binding, the binding of the acetylcholine to the receptors, gated ion channels are open. This allows sodium and potassium to pass. Okay, this is an important step. The whole point, the whole purpose of acetylcholine coming down and attaching to these receptors is to open these gates. So now that acetylcholine has fit into the receptors, the muscle fiber starts breaking it down with its special enzyme. That triggers sodium and potassium channels to open. Then last bit for this one is more sodium enters the cell compared to the amount of potassium that leaves the cell. The net result here is depolarization of the sarcolemma. What the heck does that mean? Okay, remember, sarcolemma is just our fancy word for the membrane around the muscle fiber. Depolarization is the change of charge inside the cell. So notice that sodium has a positive charge and potassium has a positive charge also. Because these ions are moving around, and a lot of them are moving around all at the same time, that changes the charge, the electrical charge inside the muscle fiber. So we have a ton of sodium coming into the cell, and we do have some potassium leaving. But so much of that positively charged sodium is coming in, it makes the inside of the muscle cell much more positive. That is called depolarization. That depolarization is what is going to allow the action potential to physically enter the muscle cell. So that's why that's important. I'll summarize this again, but questions for now? Okay, so in this first step, this is how the message, the action potential, reaches the muscle. Step 1, the action potential travels down the motor neuron and reaches the end of it, the synaptic terminal. Then we send out our neurotransmitter. The neurotransmitter crosses the synapse, binds into little receptor pockets at the motor end plate, starts being broken down. When that happens, that triggers the sodium and potassium channels to open. Then we get a bunch of sodium coming in and some potassium leaving. But so much sodium comes in and has a positive charge that it makes the inside of the muscle fiber much more positive. That is called depolarization. And we'll look at the next step on another slide, but that depolarization is what will allow the action potential to physically enter the muscle fiber. Okay, I'm going to show you the video clip that will go over just this part. We're breaking this up into 3 pieces. This is like step 1. And you can access, I have the links right here. So you can go back and look at these. These are excellent video clips.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Slightly more detail than we went into. Like we didn't give names to the gates, and we didn't talk about the recycling of acetylcholine. Don't worry about that part. But just great visual. Okay. Question?
I was kind of uneasy on how exactly the ion thing works. Like they can't decide to go, oh, it's time to go into the cell. They're ions, they're just floating around.
Right. Yeah. That level of detail is also something that we're not diving into. But we can chat about that after if you like. Okay. Okay, so we just did step one. Step one was the action potential arrives at the muscle. Okay, this is step two. Step two is, what do we do with that action potential? Now that action potential is going to enter the muscle cell, and basically that action potential is going to travel through the muscle cell and release calcium. That's pretty much what we're going to talk about here. Okay, so this whole slide and all the details is the action potential travels through the muscle cell and releases calcium. Okay, let's go through the steps. Step one, depolarization. So that was what we just looked at, with the sodium coming in and the potassium leaving, and that change in polarity in the electrical charge of the muscle cell. Depolarization results in the movement of action potentials across the sarcolemma in all directions through sodium channels. This action potential travels down the T tubules and occurs due to an electric gradient. Okay, so because the muscle cell has depolarized, that depolarization allows for the action potential to physically enter the muscle cell. The action potential enters the muscle cell through those hollow tubes we looked at on Tuesday, the T tubules. You guys remember those? They're wrapped around the myofibrils all the way across the muscle fiber. So that little bolt of lightning, our message, enters the T tubules and travels all throughout the muscle fiber at the same time. Okay, after propagation, sodium channels close and calcium channels open, restoring the resting potential of the sarcolemma. This step means after propagation is after the action potential has traveled throughout the muscle fiber. That's all that is. It's sent its message. After that part is done, those sodium channels are going to close, and the potassium channels open. And what that does is allows for that muscle fiber to regain its normal resting potential. So it's not super depolarized anymore. It's going to go back to its regular charge, its normal state. Next step, when action potential reaches the SR is the sarcoplasmic reticulum. On Tuesday, when we looked at the SR, this was that lacy blue wrapping around the myofibrils. Oh, you can see it in this picture here. That blue lace, again, is our SR. When action potential reaches the SR cisternae, so the expanded portions on the side, voltage-sensitive receptors cause the release of calcium ions into the cytosol, also known as sarcoplasm, which diffuse into sarcomeres. Okay. So as the action potential travels down those T-tubules, right next to the T-tubules on either side, we have expanded chambers of the SR. That's called the cisternae. Those expanded chambers contain a ton of calcium. There are voltage-sensitive receptors. So there's receptors that sit in the cisternae that can sense when an action potential travels down. And when the action potential comes down, the voltage receptors say, ooh, we've got a message. Open up the gates. The gates open, and it allows for calcium to be released. When calcium diffuses into the sarcomeres, this means that the calcium is able to enter those functional units to get to where it needs to go. This occurs at something that's called the triad. A triad is a cisternae on either side of a T-tubule. So you have cisternae, T-tubule, cisternae, 3 pieces is the triad. And when I say a ton of calcium, I mean 40,000 times the regular amount of calcium that you would find on the inside of the muscle fiber. Okay, so this step, the action potential has traveled down the T-tubule, has caused the opening of the gates of the SR to release the calcium. Last step, we've talked about this a little bit, calcium binds to troponin, changing its shape, weakening the bond with the actin in the other subunit. The whole purpose of getting the calcium released from the SR is so that it can bind to troponin. So that troponin can change its shape, pull tropomyosin out of place, and expose that binding site on actin. So recap on this one here. This whole step is, what do we do with the action potential once it's reached the muscle? So that action potential, that signal, enters into the muscle because the muscle fiber has been depolarized. This depolarization allows for the action potential to travel into the muscle cell, the muscle fiber, through those hollow tubes, the T-tubules. As the action potential travels down the T-tubules, it triggers gates to open in the SR. The SR is where we store the calcium. So when those gates open, a ton of calcium releases from the SR and floods into the sarcomeres. Why do we want it in the sarcomeres? That's where the actin is. So that calcium can bind to troponin, change the shape of troponin, and pull tropomyosin out of place. So in a smaller nutshell, the action potential travels into the muscle fiber in order to release calcium. Questions on this part? Still with me? I'll show you a video clip on this one.
Calls student like me, especially, you need Grammarly in your life. Grammarly is a digital writing assistant.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Sure. Okay, so calcium, very important for muscle contraction. Now, this last part here, I've actually said a few times, so this might sound a little familiar, but repetition is good for memorization, so that's okay. This last piece is basically, great, now we have calcium. What does that have to do with muscle contraction? So you do know this. So step one, when troponin changes shape and position, it pulls tropomyosin off the active site, same thing as binding site, on G-actin. Okay, so we know this. Troponin, three little yellow balls here. Troponin has the three subunits. One of those subunits is just for calcium. So calcium comes in, binds to troponin. Troponin changes shape. Because troponin is also bound to tropomyosin, when troponin changes shape, it pulls tropomyosin off of those binding sites. So the binding sites are shown here in green, and you can see this arrow is indicating this tropomyosin is being pulled off of the binding site. Next step, once the active site is exposed, myosin head binds and pulls fin filaments towards the center of the sarcomere, cross-bridge formation. We talked about this part already. So once that active site, that binding site is exposed, myosin is now able to bind to actin. When myosin head binds to actin, it forms a cross bridge. Then we go through our steps here, which we looked at the end of the lecture on Tuesday, is the sliding filament theory, is how exactly do actin and myosin produce contractions. After that cross bridge has been formed, we have the power stroke. This is when the myosin head pivots and pulls actin towards the center of the sarcomere. I'll show you a video clip after this. Then the cross bridge has to detach so that myosin can reattach and pull actin towards the center even more. We can only have myosin detach if we have enough ATP available. ATP is our body's currency for energy. So this is why using muscles takes energy. It's partially why using muscles takes energy. Because we need ATP available for myosin to be able to detach and reattach and continue to pull on actin. So here's ACE again. That's an enzyme. ATPase in the myosin head hydrolyzes, fancy word for breaks down, hydrolyzes ATP into ADP, and P is phosphate, which provides the energy needed to return myosin head to the cocked position. So this is just saying that as long as we have ATP available, myosin can continue to detach, reattach to actin, slide it towards the center, break down the ATP, and keep going. Yes?
And just, I don't think you said it, the ATP is adenosine triphosphate.
Yes.
ADP is adenosine diphosphate, so it only has 2 phosphates.
Yes, exactly. ATP, triphosphate, 3 phosphates. When ATP is hydrolyzed, is broken down, it breaks down into ADP for diphosphate, and that phosphate would have been the third one on ATP, but it's broken. Okay, does this sound a little familiar from Tuesday? Slightly? Okay, good, got some nods. Great, that's a great sign. All right, the video clip I'm gonna show you is the one that we looked at on Tuesday about the cross-bridge cycle, but it's excellent.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, The cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so now I get to share with you one of the dirtiest analogies in anatomy is the interaction between actin and myosin, with myosin wanting to bind to actin, but at rest they can't because tropomyosin and troponin are in the way, right? So essentially, it makes tropomyosin and troponin basically the cock blockers of muscle contraction because they're in the way of the binding site between myosin and actin. So that might help you remember. All right, so what do we need for muscle contraction? First thing is we need stimulation from CNS's central nervous system, brain, spinal cord. After that, we need calcium. Calcium has to come in and get the cock blockers out of the way. And then we need energy, we need ATP, because that allows for the myosin head to detach and then reattach, to continue to have that muscle contraction. How much tension we're able to produce depends on the tension that's produced at the muscle fibers, so each muscle cell, each muscle fiber, and how many muscle fibers are stimulated. We're going to expand on that in the next few slides. What we just went through, that was the super hard part. So now this is more bigger picture here. Is when we are using our muscles and we are producing muscle contraction, we have varying amounts of how much tension we're able to produce. We're looking at that in lab this week, right? So what accounts for that tension production in a muscle? One thing is how much overlap is there. So the arrangement within a sarcomere, we have to have that right amount of overlap between actin and myosin. If there's not enough overlap or there's too much overlap, there won't be the same amount of cross-bridge formation. Beyond that, we also look at the frequency of stimulation. Frequency of stimulation is how frequent, how many times are we receiving an action potential from the nervous system? One quick action potential, one single signal is going to give us a twitch. A twitch is one round of receiving a stimulus, muscle contracts, muscle relaxes. End of story. That is going to produce some muscle tension, but the lowest amount. The more frequent we receive action potentials, the more force we can produce. So the next amount from a twitch is called treppe. It's like a staircase. Treppe is when the muscle is stimulated. So we have our twitch, we have our contraction. The muscle relaxes, and then right after the muscle relaxes, we get another action potential, another stimulus. Receiving another stimulus right after the muscle relaxes allows us to actually build slightly on the tension from the first stimulus. So this shows us that if we're getting more stimuli, more action potentials, we can build up our tension. Then we move to what's called wave summation and incomplete tetanus. So here, we're actually receiving the next action potential before the muscle has relaxed all the way. So instead of completing that relaxation phase, the muscle only starts to relax. Then the next stimulus comes in and we build much more on the tension produced. And what gives us the most amount of force production is complete tetanus. Complete tetanus is when we are receiving such high frequency of action potentials that we don't start the relaxation phase at all. We just build and build and build on the tension. I think this graph here will probably clear it up for you. So here's twitch. One single stimulus gives us one contraction, one relaxation. Then we have trepe, which is like a staircase. We're building slightly. So we have one stimulus and relaxation. That second stimulus comes in right at the end of the relaxation phase. And you can see that each wave afterwards builds slightly on the one before. If we get our second stimulus in before the muscle completely relaxes, then we can build tension greatly. And if we're receiving action potentials so quick that the muscle never starts the relaxation phase, then we're able to build up the most amount of tension. Takeaway point here is the more frequent we are receiving the action potential, so the more the muscle fibers are firing, the more tension we can produce. So when we are firing a signal, that signal starts in the brain, travels down the spinal cord, out through the motor neuron. Now I don't have to say nerve, I can say motor neuron. That signal is going to innervate a group of muscle fibers. So the motor neuron plus all of the muscle fibers that motor neuron talks to is called a motor unit. A motor unit is one motor neuron, the nerve cell, that the action potential travels down. So one motor neuron plus all the muscle fibers that motor neuron innervates. It's a motor unit. A motor unit can be very, very small. If we're looking at a small muscle or a muscle that makes very fine, precise movements, like the muscles in your eye, for example, the motor units can be very, very small, where, for example, one motor neuron only innervates one muscle fiber. So we need to make very precise, controlled movements. Or we can have motor units that are very large. If we're looking at very large muscles, like muscles of the lower body, we can have one motor neuron that innervates many, many muscle fibers. So the size of the motor unit will vary depending on what muscle we're looking at. And what we just saw with that graph, force production depends on how often we are receiving that action potential from the nervous system and how many motor units we are recruiting. So both of those things combined is what makes us depend on how much force we are producing. Does that make sense? Let me give you an example here. Here's my clicker thing. And I'm gonna go pick up my clicker thing. And I know that it's really light. So when I go to pick this up, I'm making an adjustment in my brain to only activate just a few motor units, because I know it's really light. So I'm really not gonna need a ton of muscle fibers to pick this up, because it weighs like nothing, right? Okay, well, let's say for whatever reason, it's extremely heavy, and maybe it's 50 pounds. So I go to pick this up, or you can think of a time where you knew you were going to lift something heavy and you kind of like, all right, let's do it. What you're doing is you're actually recruiting more motor units. You're saying, all right, this is going to take some more muscle fibers. So like, let's get some more on board here. And you do this thing, and it's like, oh, okay, all right, I got it. I'm going to pick it up. Now, funny thing, what happens if I think that's the 50-pound one, but someone switched it out and it's the really light one? So mentally, I'm geared up for like, okay, this is going to be heavy. I got it. And I go to pick it up, and it goes flying across the room because I recruited so many motor units. I produced way more tension than was necessary. I think I actually just talked about this. So when you are going to do any sort of muscle action, you are going to recruit the correct amount of motor units. Now, generally, this starts off slowly, where you are recruiting a lower amount of motor units to begin with. And as you need more, you sort of go through this recruitment process where you're recruiting more and more and more as you go through that motion. Motor unit recruitment. Your resting muscle tone is dependent upon your muscular fitness level. So some of your motor units are always active. We talked about this a little bit on Tuesday when I was saying how you're not falling out of your chairs. So even though you don't feel like you're actively clenching your muscles, obviously there are some postural muscles that are working to various degrees. That's your resting muscle tone. So poor muscle tone would be, I have a little picture, poor muscle tone would be like a floppy little baby, where at rest, there's not a lot of latent motor unit recruitment, but there's varying degrees of resting muscle tone. So someone with a very high resting muscle tone is going to appear to have like firm muscles even when they're not actively flexing. So this would be like, in your mind, you can think of a bodybuilder that looks like they're flexing even though they're not, and that's very high muscle tone.
I was just wondering what the relationship is between muscle tone and strength.
I think I have it on here. Heightened muscle tone accelerates recruitment. So when you have high muscle tone, you are like primed and ready to recruit more muscle fibers than if you do not have as high of resting muscle tone. So because you are primed and ready to recruit more muscle fibers, right off the bat, you have the potential to produce more muscle tension than someone with low resting muscle tone. That's because of the motor unit recruitment. So resting muscle tone is important to stabilize yourself in your position, to help prevent injuries if someone were to suddenly move you. That resting muscle tone can keep you in position and protect your body and absorb shock from impacts. All right, I'm keeping an eye on the time here. We're definitely going to go up until the end of class, and whatever we don't finish, we'll just do next time because I really wanted to take our time on all this stuff before. This is pertinent to lab this week. So the different types of muscle contractions that we see, we can break down into two large categories, isotonic and isometric. Isotonic muscle contractions change the length of the muscle in some way. So within the category of isotonic contractions, we have either concentric or eccentric contractions. So if I am picking up a bag of groceries and for whatever reason I feel like doing a bicep curl with my bag of groceries, this shortening phase is the concentric phase. And then I go to set down my bag of groceries, and I'm just going to again, for whatever reason, I'm just going to set it down like this because I'm strange. And this is the eccentric phase here, is muscle lengthening.
But they're both called contraction.
They're both contractions, certainly, because when you are slowly and controlling this movement down, your muscles are definitely still working. I'm not just letting the bag fall. I am controlling it on the way down, and I'm doing that with my muscles. That's eccentric. Then we have isometric. An isometric contraction is when your muscles are definitely contracting. You're not producing any movement, though. So if I were to come up here and try to pick up this table, and the table weighed 200 pounds, and I'm really trying to pull it, my muscles are definitely working. I can feel my muscles working, but nothing's moving. That's isometric.
Another isometric example would that be like a
Plank. Yeah, plank is great. Yeah. Okay, energy reserves. There's actually a ton that we could say about energy reserves. We get into this more in is the digestive system. In 371, we get into this quite a bit more. This barely raises the surface, but we will just note that. We know we need energy in order to produce muscle contractions. There are different areas that can supply us with energy for these exercises. So the first thing that we do when we're using our muscles is we're going to use whatever available ATP is already present. That only really gives us about 2 seconds worth, though, of activity. After that, we get into the creatine phosphate system. That gives us the next 15 seconds or so of energy. Now, if you are a world-class sprinter and you're doing the 100-meter dash and you're doing it in less than 15 seconds, then you don't need anything past the creatine phosphate system. So if you only need a very quick burst of energy, maybe that's as far as you go. But if you're doing something that perhaps lasts more than 15 seconds, after that we get into glycolysis, where we use glycogen. That gives us the next, say, 2 minutes or so worth of energy. This process, glycolysis, is anaerobic. Anaerobic means we do not have to use oxygen in order to produce that energy. That's also why it's not long-lasting. But if you were doing, again, something that didn't last a very long time, perhaps glycolysis would be enough. After that, then we start getting into aerobic metabolism. In aerobic metabolism, we rely on oxygen in order to create energy. I'm not going to go into any more detail than that because there's a lot of detail, and I'm just going to say the longer the exercise goes on for, as far as time, the more we rely on aerobic mechanisms. Okay, so we know we need central nervous system stimulation, calcium, and energy for muscle contractions. And that should be it, right? We should be able to just go forever if we have those 3 things. No, we know that's not true. We've all felt that feeling of, I am done for. I have done too much. I am done for. What exactly is happening when we are fatiguing our muscles? We could be depleting our metabolic reserves. So that would be a various compilation of things that we need to sustain the muscle. So we've run through the amount of glucose that's available, for example. Or there's been some damage to the structure of the muscle fiber, to the membrane, the sarcolemma, or the sarcoplasmic reticulum. When we exercise, our body heats up, and there's also a change in our blood pH. That change in pH can throw off the balance and reduce how much calcium can bind to troponin. We know how important calcium is, So if we have reduced calcium binding, that's also going to throw off our muscle fatigue.
The pHs, those are lactic acids being used, right?
And then psychological fatigue. I know that I've definitely felt this one. For some reason, I decided to run a half marathon like years ago. And at some point, when you're just running forever and you're thinking, oh my gosh, everything hurts. Why am I doing this? Psychological fatigue. Like, I really want to stop. And that's definitely a component. And the effects just accumulate. They build on top of each other. It's not just one thing. It's usually several things put together. And then we get that feeling that we have to stop. But this is what's going on. Okay, I want to do this justice, so we'll call it there for today. We'll finish up. We'll start here next time, and we'll finish from there. Stop here.